as president of SASBi, it is a tremendous pleasure to have Tulio come and talk to us today. His topic is particularly apt since it is about COVID, and we are holding this virtually because of that. So first, I'd like to say that Tulio hails from Brazil, and he got his bachelor's degree from the Federal University of Rio Grande do Sul, which is in the province or state, actually, Rio Grande do Sul, which is Great River of the South. It's the southernmost state of Brazil, sort of lies there just between Uruguay and Argentina, or just above Uruguay. Anyway, so he moved to South Africa in 1997, got his master's degree and PhD degree from the Nelson R. Mandela School of Medicine and UKZN. Somebody is saying that you can't hear any sound. Is that better? Sound is working. Good. Sorry to interrupt with that. So then Tulio got a fellowship. It's the European Commission's Marie Curie Research Fellow, and he was at the University of Oxford, and that was 2004 to 2006. Then between 2019 and 2015, he was director of the genomics program at the Wellcome Trust Africa Center for Health Populations and Population Study in South Africa, became an affiliate senior lecturer at the University College of London, got another very important fellowship, which is the Royal Society Newton Advanced Fellow at the Wellcome Trust Sanger Institute in Cambridgeshire, and was also at the University of Edinburgh, apparently. But in 2015, he became a full professor at the UKZN, was elected president of SASBi in 2016, founded CRISP, which is the KZN Research Institute, in 2017. In 2018, he became associate professor at the University of Washington, Seattle. And since 2021, July, he's a faculty member and professor at Stellenbosch University with appointments in the Faculty of Medicine and Health Sciences, as well as the School of Data Science and Computational Thinking. And his topic of interest has always been viral outbreaks, viruses, virus genomics, and viral outbreaks. And he's interested in HIV, hepatitis, chikungunya, dengue, SARS-CoV-2 now, Zika, and yellow fever viruses. He has more than 180 publications in PubMed, many of them in the top scientific journals such as Nature. And according to Google Scholar, he has an H index of 51 and I-10 of 155. So we welcome Tulio, scholar and past president of SASBi, a new faculty member of Stellenbosch University, to inform us about his research, and his talk is titled COVID-19 Genomics Surveillance in Africa. And just a footnote, Tulio has got a plane to catch, so he will be ending his talk at 11:40. Welcome, Tulio.
Yes, thank you, Herod, and hello, colleagues from SASBi. Yeah, as Herod highlighted, I love traveling, I love collaborating, and I love working across many different universities. To be honest, that's one thing that is phenomenal about bioinformatics, is that you can collaborate with lots of people, yeah, and it's quite exciting to see how a lot of times the same tools that we use for a pathogen can be used through another one. So as Herod said, yeah, I will be presenting today about SARS-CoV-2. Many of you may have heard a little bit about our work on SARS-CoV-2 because it has been very well publicized, not in the scientific only, but also in the general media. Yeah. And just to highlight that the main thing why we could respond so well, at least with genomics and bioinformatics in South Africa, is what Harold Trump highlighted, that we have been working for the past 10, 20 years in almost every big viral epidemic that happened in the world. Yeah. And in the end, the process is quite similar. Yeah. So today I'm going to be doing a presentation. I'm going to try to take half an hour and 10 minutes for questions. Then I'm jumping a plane to come to Stellenbosch. Yeah. So what I'm going to be highlighting, it's something that in the end we found really, really interesting. Yeah, how so much change between the first, second, and third wave in South Africa, especially in viral genomic diversity, which gave rise first to lineages and then variants, and each time a different one to begin to dominate. Yeah, I'm going to show that as we work with many colleagues in Africa, we just got a paper that is accepted in Science with 120 different groups in Africa. We have identified together with then many, many variants in Africa. Yeah, and then we're going to highlight how do we think that this variant emerge and why we have to be very careful in places such as South Africa or Africa do not become a factory of variants. And then I hope to take some questions and answers. Yeah. So just to highlight, yeah, and I think in the introduction from Harold, it was quite clear that we do like working across big networks, across big groups, in which a lot of time involved me moving between organizations. Yeah. So very early in this pandemic, we set up the Network for Genomic Surveillance in South Africa, or NGSSA, and that is between 8 different sites, yeah, including WIT, University of Pretoria, CRISP at UKZN, the University of Free State, University of Cape Town, Stellenbosch, and the NICD. And then what you're going to see that all of these sites, yeah, which can produce genomic in real time, they are always close to a green box that is the National Health Laboratory Service, just allowing samples to flow very quick and to be genotyped. Yeah. And because this was a true group effort, I'm going to be showing in every single slide the logo of all these fantastic collaborators that are working together. Yeah. So just to highlight, I don't need to remind you, but because a lot of my presentation is going to focus on this difference between a first wave, second wave, and third wave. Yeah, but not really on cases, deaths, and restrictions, but what happened in each of the wave with viral diversity. Yeah, because that's what we like analyzing is viral genetic diversity using bioinformatics tools. Yeah. So just to ground you here, it is in this panel A, we are just looking at the first wave. The first wave in South Africa started quite early March. Yeah, here is case in black. In red is the reproductive number, which normally is above 1. We know that's growing. When it's below 1, we know that the pandemic will be decreasing. And here in the little blue is the genomes that we were sequenced during that time. So we have been quite fortunate in South Africa and starting very early. To be honest, we sequenced some of the first genomes in March and April, even a contact of the first traveler that was identified in South Africa. And what we found here, if you put here in the x-axis in the panel B, let's just get the laser. Yeah, in the x-axis time, the y-axis, it is just count, and the count is number of genomes that we can identify the origin. Yeah, and what you're going to notice is that the majority of our introductions into South Africa, it happened between February and May. And the main color here is the brown, which it means it's from Europe. So 80% of our introductions in South Africa of SARS-CoV-2 in the first wave were from Europe. Yeah. And why Europe? Yeah. Europe is, yeah, just 1 second. Yeah. And Europe, sorry, someone was just being loud. And Europe is because 80% of our air traffic was from Europe, it come from outside the African continent. So the epidemic was seeded from Europe, but then the same information here is in the panel C in black. So what it means, we have many, potentially hundreds of seedings of infections, but very quick, the majority was being transmitted locally, which is within South Africa. So very quick, the introduction started getting transmission chains. Yeah. So that was a paper that we published in Nature Medicine, and as the name highlight, in total, as we discriminate the South African variation, we have identified 16 unique lineages in South Africa that dominate the first wave. Yeah. Just to understand how we do that, here is what you call a clock phylogenetic tree. Here in the x-axis is the date of when there was sampling. December is 2019, and here's 2020. It was the first wave. We had 3 different phases in South Africa. We have the early introductions, we have the emergence of lineages, and then we have peak infections, which were mostly characterizing of lineages that were introduced, but it began to become South Africa-specific lineages. What had happened? Normally, when you go to an amplification event, such as an outbreak, yeah, the virus, they fix 1 or 2 mutations, and they become the unique lineage that differentiate them from the rest of the international virus, yeah. So we have identified 16 lineages in South Africa, but we have identified 4 of them that were very high prevalence and 3 of them that were dominating the first wave. And in the classification of SARS-CoV-2, they get these funny numbers and names, yeah. So the 3 lineages that are going to be described is the B156, the B.154, and the C1. And these lineages, what they did, yeah, not only we could identify the mutations then, but we could map them in space and time. So, for example, the B156 was a lineage that dominated a lot of the infections, or at least 1/3 of the infections in KwaZulu-Natal. Here, when I have in blue is the date of origin, going back to the past, and in red to the present. So this one was potentially emerged in Durban, very quick went to Richards Bay, and then started being introduced in all the districts. So you had what we call regional lineages, and then you had national lineages. So, for example, the C1 emerged in Gauteng, yeah, which caused a big outbreak in a hospital in the northwest. And then, very interesting, to KwaZulu-Natal, it moved across the highway. As you know, many people, it's a very busy road that comes from the harbor to Johannesburg, and went introduction in the northwest of the province until it started spreading in all the different districts. Yeah, so that is lineages. What it means, they are different. They have 1 or 2 mutations that differentiate them, but neither of them are mutations that increase very rapidly transmissibility. So what it means with them? Yeah. So if you just go here, we're going to go a little bit more recent time. The axis is the date. Yeah, here is going 2020, 2021. With the y-axis is the proportion of the genome. When I have a white one, normally I have 10, 20 lineages. Yeah. And here is the prevalence of the 3 first lineages that I described, the B154, B156, and 61. So what we see that they co-circulate in South Africa, but never at very high prevalence, because they didn't seem to have main advantage for the transmissibility. And everything changed around the end of last year, that you have a new lineage that starts to completely dominate and displace all the other lineages. Initially, we named that 501v2, but that was get to know as the beta variant. Yeah. What is different between a variant and a lineage? So a variant, now it has many mutations. So this variant has around like 24 different mutations that separate in the genome of the virus, and mutations where you don't want that to be. More specific, here in green is the spike glycoprotein, yeah. And here is the different areas of this glycoprotein. So this one has 3 mutations in the receptor binding domain, including the 501Y, which increases transmissibility, and the 484K that decreases neutralization of vaccines, but I'm going to get more details. Yeah. So once you add the mutations both in the spike receptor binding domain or close to the furin cleavage site, that normally allow the variant either to transmit much faster or start escaping from neutralization. Yeah. And I will bring back some of the other mutations after. We're going to talk a little bit more 501, 484, the 417, but also these deletion regions which are in the antigenic supersite. Yeah. So now we had like a lineage in asteroids, which became quite a famous variant. Yeah, not only it emerged in the Nelson Mandela Bay, but very quick. We are talking within like 2 to 3 months, it spread everywhere in South Africa, especially across the coast, and displaced all the other 16 lineages that we have identified. Why that? Because this variant was much more transmissible, and that's why the second wave comes so fast and so deadly. Yeah, and that one spread a lot across the coast, and then after went inland, and the third wave coming the opposite way. It came from inland to the coast, but I will get in detail now. Yeah. What this beta variant or the B1512? What we did some work a lot with Shabi Ahmadi. He sent all the samples to be genotyped in our lab. And we work with 2 colleagues to try to answer one big question. Does plasma collected from people after receive what was the main vaccine that was being rolled out in South Africa, the AstraZeneca or the Chadox, neutralize this variant? And the answer, it was very poorly. Yeah. So for example, the beta variant, which also known by B1351, of around 75% of the plasma that we put from people that were vaccinated against a pseudovirus or virus assay was a completely knockout, very little neutralization. So neutralization was strong attenuated, yeah, and in a high percentage of them, depend of the assay, you have a complete knockout. And that was the reason why South Africa did not roll the AstraZeneca during the second wave of the epidemic. Yeah. What we also do with a colleague of mine, Salim Abdul Karim, we keep an eye not only in all the results that come from pseudovirus and live virus, and which allow us to try to work on either decrease of neutralization with the beta variant, decrease neutralization of all the vaccines, but some of them much higher, and I try to together with clinical trial to try to identify which vaccines were more appropriated to South Africa. When we put that with clinical trial that exists in South Africa, the AstraZeneca not only had very little neutralization, but very low efficacy about clinical moderate. Unfortunately, it was a small trial and didn't have data in clinical efficacy, moderate and severe, but another vaccine had the Johnson & Johnson, and that was very effective against severe hospitalization and disease against the beta variant. And the second other vaccine that data existed was a smaller decrease of neutralization in the Pfizer, and that was one of the main reasons why South Africa chose these 2 vaccines to be rolled out initially, because they were very efficient, very potent, and very good against the beta variant. Yeah. And we really thought that was the end of our variation in South Africa. Just to ground you, we have here the axis come to very recently now, the y-axis proportion of the genome. Here in gray is when you had the 16 lineages, the beta variant dominate, and then we have introduction of other 2 variants of concern, the alpha that never went to dominate, but then the delta variant that not only dominate but displace a already much more transmissible variant. Yeah. What we did is to work that quite quick. So for example, the first province that identified the growth of the Delta variant was KwaZulu-Natal, and was our group that did that. So what we see is that as it grew the prevalence in green of the Delta variant, which was displacing the Beta variant and other lineages, you had this exponential growth of cases. Yeah. At the moment, we just finished the analysis and the paper that the Delta replaced Beta, but what it seems is that Delta is around 30 to 40% more transmissible than Beta, and Beta was already like 50% more transmissible than the other lineages. So what we are talking here, we are talking about a variant, the Delta, that at twice more transmissible, at least, than the normal virus that caused the pandemic. Yeah. The same was seen with a little delay, but also in Johannesburg and Gauteng. And this graphic is repeated in all provinces, including the Western Cape, the Free State, the Limpopo, the Northwest, the Eastern Cape. Always what we see in this third wave, increase of frequency of the Delta variant, very, very fast increase of number of cases. Yeah. What this Delta variant have, we're quite surprised. The first, it has much less mutation than the Beta. The Beta has around 13 mutations in the spike region. That one has much less. Has only 2 mutations at the receptor binding domain. Okay, but it has this very nasty mutation that's exactly at the furin cleavage site, yeah, which is a mutation from a proline, a P681 to a R amino acid, and that is what the scientific believe the main mutation that make this variant extremely more transmissible. If you look at where it is in the surface, these mutations, they will be normally exposed in the surface, yeah, especially because this normally is caused by neutralization that was not very effective, yeah, from antibodies, which allow them to create a mutation, and if they increase the peak fitness, they will become more transmissible. What's happened with Delta? Yeah, I'm not gonna get in detail a lot because I think that more exciting is what I'm gonna be talking next. Yeah, but what's happened? We have many other variants that have emerged in Africa. Yeah, and the one that became the more famous was the Beta one, as I described, but we also have a C1 in South Africa that became the C.1, which had a mutation, the 681H, yeah, and now we have the C.1.2 that had a lot of mutations, in which I describe now with the NICD. We had another variant that dominated the whole second wave of Uganda, Rwanda. That is the H23.1. Again, characteristic, look, the same mutation, the same guy coming here, the 681R of the Delta variant. And Nigeria, it also have create their own variant, which they have this mutation that we worry a lot, the 484K, yeah. So what we have, it is that variants, they keep emerging quite often in Africa, yeah, which then they cease to exist when they are less transmissible, but we have a large number of variants emerging in Africa, including another variant that we have described with the Ministry of Health of Angola from travelers from Tanzania. This is a variant of the A lineages, one that we almost thought that would be extinct, yeah. And this variant was a big jump in evolution, yeah. And that one has close to 40 mutations, yeah, almost the double of the beta variant, yeah, and a lot of the mutations in the spike gene. So what we are seeing is this emergence of these variants with lots of more mutations, but normally similar mutations. The 681 increased transmissibility, 484 that decreased neutralization, and some of the deletions. So the virus is going to a continuous evolution in the African continent. Yeah. So one question that we are trying to really answer is how do SARS-CoV-2 variant emerge? Yeah, so we know that it's by 2 different processes. It's a gradual accumulation of mutation during community transmission. This is what causes a lot of the initial lineages. Each time that you have a large outbreak, you're gonna transmit one individual to all, and then they will catch that mutation in that individual and become prevalence. But what's become clear is that also to a process of accelerate intra-host evolution during prolonged infection, and that's where we are worrying a little bit for Africa there. This is not only common in the African continent. The first case was published last year, and that was a SARS-CoV-2 evolution in an immune-compromised host that was in Boston, in America. What it was is a small image, but I'm gonna try to explain here. Was an individual that was infected. He kept like close to 150 days infected. This y-axis, it is the CT score. Okay, which what it means, once he have a CT score above 40, he has virus, he cannot clear the virus. And what's happened with that virus, they sequence all the time points, and this virus create large number of mutations, yeah, And many of the mutations of the variants, for example, it may not be able to see clear, but here the 501Y, that was characteristic of the beta variant, yeah. And this individual could not clear the virus, was a cancer patient, yeah. And what he was receiving is also immune-based therapy or in convalescent plasma. So we knew that immune-compromised individuals, especially from cancer, can not clear the virus and generate mutations. So, for example, the alpha variant of the UK also come from one of these individuals. But recently, we described a clinical case in South Africa of a 36-year-old HIV-positive female, which presented just before the second wave with sore throat, cough, and dyspnea. She was on prior antiretroviral therapy, but she has disengaged from care, really bad adherence, yeah, and she had a very, very small number of CD4. So this is a severe immune-compromised individual with CD4 is staying too close to 200 days at single digits, yeah. She has a very high viral load, yeah, and then the symptoms is that she presents with respiratory distress with a rate of 30. She tested positive for SARS-CoV-2 with a low CT score. What it means is there is a lot of virus. She cannot clear SARS-CoV-2. She's administered oxygen via face mask and a cortisoid, the dexamethasone, and she's enrolled in a prospective study exploring immune response to SARS-CoV-2, which we collaborate with Professor Alex Siegel from ARI, yeah. So she kept coming back, and we had 210 days of follow-up of that individuals, yeah. And just highlighting one time point that's become very important, 10th of December last year, when the clinicians managed her, switch her to second line ART, yeah. So it switched the antiretroviral therapy, but she had very big problem with tolerability. She arrived with very complaint of chest tightness, and she desaturate very, very fast on exercise, really showing that she's beginning to develop potentially some disease. But then on the X-ray, the lungs are quite clear, and she refused hospital admission, and she's just treated as an outpatient of pneumocystis jirovecii pneumonia with sorry, I find difficult to talk the drug that she was, and then she recovered. But then, as she recovered from this treatment, she stayed fully asymptomatic during follow-up. Yeah, and that's again the very important collaboration that we have with clinicians in case. Why this case was really interested, at least evolutionary, yeah. Here, just to highlight to you, in the X, in the X, I have viral load, HIV in blue, and in the Y is an inverted of SARS-CoV-2 CT score, which it becomes similar to HIV viral load. That individual, when she presenting on day 6, she's not suppressed for HIV. Each time that we sample her on days 0, 6, 20, 34, 71, 106, one night, she's still unsuppressed for ART. We sequence both the HIV and the SARS-CoV-2 virus. The HIV virus have very high level resistance to the current treatment, and then she switched to effective treatment and managed to suppress the HIV virus, so the immune system begin to become functional because she's on ART, and the same happened with SARS-CoV-2 with a 2 weeks delay. So only at 233, this individual it is 230 days that she could not clear the SARS-CoV-2 virus. Yeah. So what we did is get access to nasopharyngeal swab and sequence every single time point, and what we see this very big virus evolution. The first thing, we are even worried that the samples have been mixed up because they had so much mutation, but when you put in a very large phylogenetic tree, they all cluster together, so it's quite clear that's an intra-host virus evolution. And that virus that we sequenced in between October and April 2021, start adding many mutations. So here's day 0, day 6, day 20, day 30, and the mutations it's shown here in the internal branch lengths. So what we can see with that individual is the evolution of most of the common mutations that we see in the variant, understanding that this individual, here is the variant, the beta variant, and the delta variant was not a variant, but was an evolution inside the individual, adding many of the mutations. The deletion is found in many variants. The 484K decrease neutralization is found in many variants, and both the 490 and the 501. So what we could see is this completely virus evolution in an immune-compromised individual due to HIV that add most of the mutations of the variants of concerns of SARS-CoV-2. Yeah. So just to conclude, what we have seen during this program of genomic surveillance in South Africa and Africa, we have seen multiple variants have emerged. We can count up to 10 different variants and hundreds of lineages. Yeah, we have now good evidence that prolonged infection in immune-compromised individual is one of the mechanisms for the emergence of SARS-CoV-2 variants. We have demonstrated multi-mutational escape variants in an individual with advanced HIV and prolonged ART failures, which then the SARS-CoV-2 replications terminated short after introduction of effective ART. So now we are discussing what's the clinical implication. So for example, we believe that all people with COVID-19 should be tested for HIV and should be on effective ART, otherwise the regimen has to be changed, otherwise they have a chance that they cannot clear the virus and the virus can accumulate large number of mutations. And we really need to control the pandemic, the SARS-CoV-2 in the continent, because we have the largest immune suppressed individual population in the world, and we don't want to see that we really, our fear is that immune suppressed individuals in Africa could become a kind of factory of variants. So it's a crucial importance to increase vaccination and try to control the pandemic as soon as possible. Yeah, thank you, Harold. Yeah. And this, as I highlight, is the work of hundreds and hundreds of scientists of multiple organizations in South Africa. And one thing that we have shown in South Africa, it is if you can get many different university and government labs to work together, we can answer some of the questions quite quick for this pandemic. Thank you very much. Yeah.
Thank you, Tulio. That was a great talk. I will open the floor to questions. I do have my own question, but I'll wait with that for a little while. Are there any questions? Could you please raise your hand so I can note that? Helena has a question. Please go ahead.
Hi, Tulio. That was a fantastic talk and absolutely great work. I'm just curious about the logistics. So who sends you samples? Is it the National Health Laboratory Service? And how many samples altogether have you sequenced in the past 2 years?
Okay, so who send us samples? Yeah, we have a number of different samples strategy. For example, in the provinces in KwaZulu-Natal or in the Western Cape, we run what we call a lightweight surveillance of around 50 or to 100 random selected samples from across the clinics of the NHLS. And we do that from the beginning of the pandemic because we found very effective to be doing that at random selected samples, especially. We also do a lot of vaccine breakthrough. I haven't shown you these results here. So that's when we work with Shabir Mahdi or Glenda Gray in all the vaccine at trials that we have in South Africa. Yeah. And more what we do also is to respond to outbreaks. So normally outbreak, we send an outbreak investigation team to do sampling that. So we get samples a lot from the NHLS. That's why the sequence facilities are close to each NHLS place to do, and then with samples that are important to answer a given question. In addition, now we are receiving samples from over 10 different African countries. Yeah. So the network of genomic surveillance, or our lab here at CRISP, which also is setting one now is Stellenbosch, or the NICD, we have done close to 20,000 genomes for South Africa and Africa, of like 35,000 total genomes for the continent. Yeah.
Okay, I see Rebecca Todokera has her hand up. Please go ahead.
Okay. Thank you, Tulio, and thanks, Gerhard, for hosting this talk. Very fascinating. My question is with regards to so what you've shown is the emergence of variants in immunocompromised patients. And right. So I'm just wondering, and I've thought about this for a while, I'm just wondering what your thoughts are with regards to the vaccination strategy. Should we be thinking about vaccinating immunocompromised, maybe people with comorbidities, rather than an age-based vaccination strategy to try and stay ahead of these new variants coming? Because I feel like we might be continuously trying to stop the fire here with more variants emerging if we are not very strategic in how we approach the vaccination strategy. Yeah, I'd be interested in hearing your thoughts on that. Thank you.
Okay, thank you. Yeah. I must disclaimer that I am also a member of the Ministerial Advisory Committee, and there is a VMAC, also a Vaccine Ministerial Advisory Committee. And as one review all the literature and the effect everywhere in the world and South Africa, age structure vaccination, it is the most effective to save lives, with no doubt. Yeah, because mortality and hospitalization is closely associated with age and comorbidity, but it's much, much easier to run a large-scale vaccination by age structure, because everyone knows the age. So and that's why it's so important. One thing that we know at the moment is that South Africa has now a large amount of vaccines in the country, over 10 million in stock. So potentially by next week, every adult will have access to a vaccine, and I really encourage people to vaccinate. It's the only way that we're going to get out of this pandemic, unfortunately. Yeah. In relationship to HIV positive individuals, it's a very important question, because if they are virus suppressed, they respond exactly as an infected person. The immune system is working well. If they're immune compromised, the vaccine doesn't generate large number of antibodies. We're going to be publishing that with Alex Siegel in a couple of papers in the next few weeks. Yeah, so there is a number of operational projects and trials to try to see if maybe immune compromised individuals need not only 2 shots, but potentially a 3rd shot to be able to bring more neutralization antibodies, but more important, if they are on HIV, to get on ART treatment. And since we highlight these results, honestly, we first present to our Minister of Health and even our President. They decide to increase the ART program by another 2 million people in South Africa to be put on ART. So unfortunately, we have these twin epidemics, as we had for years, HIV and TB, and we think that's more relationship between the SARS and HIV than we expect. Yeah.
Well, thank you. So my last question is we've now sequenced a lot of individuals, and we've seen a lot of evolution within individuals. Have we seen most of the variants that are going to change fitness? I mean, there's still combinatorics of increasing the combinatorics, but within the individual variants.
The answer, Gerard, is that we honestly we do not know. Yeah, we really thought a few months ago that the mutations, the 501 and the 484, were kind of the peak of transmissibility and immune evasion, until the Delta variant came without that mutations and with the 681. Yeah, we know that there is a process of convergence evolution, and there is a beautiful paper from Darren Martin coming Cell in the next couple of weeks, which we also call AUTO, that show that all the variants are evolving in a kind of a positive selection towards a fitness peak and add the mutation. But One thing that we also know, so some people believe that the virus got to the peak of the fitness, but this virus have surprised us so many times. So the really answer, we don't know, and we really hope it doesn't surprise us again. Yeah.
Okay, I'm going to give one last question to Olaitan Awe. Please go ahead.
Thank you very much. Thanks, Tulio. That was a great presentation. So my quick question is there a database or archive where these African SARS-CoV-2 variants are deposited? And if that's true, are these data sets well annotated in terms of the African country where they emerged and some other metadata that would be useful for folks to use in some analysis?
Yes, that is. The database that the scientific community use, it's very well annotated with the metadata. It's called GISAID, GISAID. Yeah, it is one need to register to get a login. Yeah, and and people can analyze the data. But one of the demands of this database is that they acknowledge the people that did the data generation. Yeah. It has 2.5 by now 2.7 million genomes, and they are very well annotated, and they are a great resource for analysis and a great resource for collaboration. That's what allows us to put this African paper with 120 collaborators in Africa, because not only we had a common database that we could work together, the GISAID, but were very well annotated, not only the authors, but also the laboratory that generate and sample the virus. Yeah.
Thank you.
So thank you, Tulio. I'm going to close the session now. Thank you very much. Thank you as ex-president, and I hope to see activity from you in SASBi some more. So thank you, and SASBi thanks you for your efforts.
Yeah. Thank you for the invitation. And let me run to get my plan, because I'm dying to come to Stellenbosch. Thank you, everyone.
Okay.